this one here we're looking at the formation of urine. So let's look first of all at the aim. Now the aim of this is to be able to describe and explain production of urine with reference to processes as ultrafiltration and also selective reabsorption. Let's just recap first of all what we've seen in the previous video. Now we looked at a kidney tubule or nephron. We described briefly what happens in various stages. So look first of all here, this topic here, this is your proximal convoluted tubule here. Okay, and here fluid is altered by reabsorption of all the sugars, most salts and some water. So about 85% of the fluid is reabsorbed at this point here. We then go into the descending limb of the loop of Henle. That's this section here, isn't it? Okay, and here water potential of the fluid is decreased by addition of salts and removal of water. Then we come into the ascending limb of the loop of Henle, which is this, oops, lost my little pointer there, which is this section here. There we go, not very tidy, but there we go. Now here, water potential is increased as salts are removed by active transport, so energy demanding section here. And the last bit we talked about was the collecting duct, which is this bit over here. Now the collecting duct, okay, water potential decreased again by removal of water. Now this ensures that the final product, the urine, has a low water potential. You don't want to be excreting more water than you need to. Therefore urine has a higher concentration of solutes than blood and tissue fluid. Right, now in this section we're going to really focusing on up here Bowman's capsule. Let's go on the next slide, shall we? Now here's Bowman's capsule and the glomerulus in structure. You've seen this before. Now what we've got here, this is a magnified view of this area here. So what we're looking at here is a magnified view of this bit of the membrane just here. Okay, so bear that in mind. Let's look now at some of the features of this. First of all, look at the diameter, and this is what's called the afferent arteriole. Now the afferent arteriole is very wide compared to this bit here. Now what that means is that you're going to build up pressure. Imagine loads of people rushing down a corridor and suddenly the corridor gets narrower. Now there's going to be an increased pressure. Now, that increased pressure is really important. So in here you get increased pressure in the glomerulus, and the effect of that is the pressure is higher in the glomerulus than in Bowman's capsule. So pressure is higher here than here, so fluid is pushed in this direction from the glomerulus into Bowman's capsule. So high pressure caused by the difference of diameter here is really important. Now here's that magnified view I talked about of the membrane. Okay, now the barrier between the capillary blood and the lumen of Bowman's capsule is made up of three quite distinct layers. The first layer is called the endothelium of the capillaries. That's this part here. Okay, now you see these narrow gaps. There are narrow gaps here, okay, on there, between the cells through which blood plasma and dissolved substances can pass through. We then got this basement membrane. Here's your basement membrane coming along here. Now this is a fine mesh of collagen fibers and glycoproteins, and this really is a filter. What it does, it filters what's passing from the glomerulus into Bowman's capsule. Sorry, that's the wrong way, and there it goes there. There we go, like that. There we go. So this filters it, and this prevents the passage of large molecules with molecular weights in excess of 69,000. So talking about, you know, we can't allow through any red blood cells and also larger proteins. The third part are the podocytes. Now these are the epithelial cells here, epithelium there, okay, of Bowman's capsule. Podo, they're finger-like projections, and what they do, they're called major processes and ensure gaps between the cells. You can see how the filtered stuff is passing between the gaps here. There we go, rushing down there. Now, most of the ultrafiltration involves the basement membrane. So this bit here is where most of the filtration occurs. Other layers do little to filter out larger molecules. Now this is what happens when the blood is filtered. So here we're going from the glomerulus here into Bowman's capsule, okay, hence the arrow is showing, going from glomerulus into Bowman's capsule. What happens to the concentrations? Well, look, all of the water as it goes through gets filtered. All water is filtered out of the blood into Bowman's capsule. Proteins, not a lot, 0.005. So this is only the very smallest proteins with molecular weights less than 69,000. All amino acids get filtered through, all the glucose, all the urea and all inorganic ions. Now a lot of this stuff is what we might actually need, therefore there's been a lot of reabsorption, isn't there? But just to explain, blood cells and large proteins are left in the capillary. The very low water potential is important in ensuring reabsorption of water at a later stage. So when your blood leaves via the efferent arteriole, it's got very low water potential, which means it can readily absorb stuff at a later stage inside the kidney. Excuse me. Now, staggering figure here, the total volume of fluid filtered out of the blood by both kidneys is about 125 cubic centimeters per minute. This represents about 180 litres a day. Now imagine if nothing was reabsorbed, you'd spend your whole day on going to the toilet, wouldn't you? So much of this stuff has to be reabsorbed. Let's look now where this happens. Now we said before, 85% of the filtrate is reabsorbed in this area here called the PCT, or Proximal Convoluted Tubule. All glucose, amino acids and some salts are reabsorbed in this area here along with some of the water. Okay, so we're getting the stuff back here. Let's look in more detail now at the wall of the PCT. Now make sure now you understand what we're looking at. We're looking at a cross section of, say, this bit here. This bit represents the wall of the PCT. This is the fluid filled space inside the PCT, the lumen, and over here we've got the capillary. Okay, so anything that's reabsorbed is going to go in this direction here. So make sure you got your bearings here. Okay, let's have some labels, shall we? Now this is the lumen of your PCT. Lumen is a fluid filled space inside your PCT. This is where all your filtered stuff is currently passing. Water, glucose, amino acids, all going down here. Now this bit here represents, this bit, okay, is the wall of your PCT. This bit is the wall of PCT. In there you've got nucleus, you've got mitochondria. This then is your capillary. Over here is your capillary. You can see you've got the endothelium here of your capillary, and you've got the lumen of your capillary, the blood plasma with red blood cells. So make sure you can clearly make out what's what in this situation. Right, let's look now at the processes involved, shall we? First feature is this thing here. Now this is the inner wall of the PCT. This is the inner wall here, the PCT, and notice how folded it is. It says here, highly folded microvilli, and what they will do, as always with villi, they increase the surface area for reabsorption. So very, very important. We've also got a code called co-transporter proteins, and they transport glucose and amino acids. There we go. So these things here are being transported by co-transporter proteins, okay, along with sodium ions. There's some ions. Oh, God, I missed out on there. Okay, from the tube into the cell. And this is called facilitated diffusion. Now, a couple of terms here. Let's just think about these two terms: co-transporter and facilitated diffusion. Now, a co-transporter protein, what they do, allow facilitated diffusion of simple ions to be accompanied by the transport of larger molecules such as glucose. So simple ions are being transported across, and in addition you get transport of amino acids and glucose. Then we've got facilitated diffusion. Now this is diffusion enhanced by action of proteins in the cell membrane. So here you've got proteins that actually help move stuff across the membrane from the lumen into the wall of the PCT. So it's two key terms. Make sure you understand the terms co-transporter proteins and also facilitated diffusion. Not sure? Go back, have a look, and really ensure please you understand it. Now over here again we've got folding of the membrane. Again we've got these microvilli here. So membrane is folded to increase surface area. Now this area also includes what we call a sodium potassium pump, and what that does, it pumps sodium out and it pumps potassium in. So sodium is going to leave and potassium is going to come in. That's really important. We'll see in just a moment. Now sodium potassium pumps are special proteins in the cell surface membrane that actively transport sodium potassium ions against their concentration gradients. Now if you are getting transport against concentration gradients, you can guarantee you're going to need lots and lots of energy, and therefore you've got loads and loads of mitochondria. So mitochondria are present. What these do, these provide energy from ATP to energize this sodium and potassium pump. Let's see now the various stages. Now, first of all, sodium ions are actively transported out of the cell into tissue fluid. So you're getting energy being used to pump the sodium out of the PCT into the tissue fluid. Okay, in tissue fluid, sodium being pumped out. Now, what that means is you've got a lower sodium concentration here. That's very useful. Now, because then glucose or amino acids enter cells with the sodium ions. Because there's now a lower sodium concentration here, sodium ions, okay, diffused in there, facilitated diffusion, okay, along with glucose or amino acids. So here there's a lower concentration of glucose and amino acids. Now, as they diffuse in, builds up the concentration of glucose and amino acids. It's higher here than it is here, so glucose and amino acids diffuse into the blood capillary as you'd expect. There's a higher concentration here, you'd expect it to diffuse this way into the blood capillary. Okay, so there's our glucose and amino acids. So salts, glucose and amino acids are reabsorbed, reducing the water potential in the cell. This therefore has a lower water potential, and surprise surprise, because this has a lower water potential, water is going to move into this area here. Okay, this increases the water potential in tubule fluid, so it enters the cells, then is reabsorbed back into the blood by diffusion. Okay, there's going to be a high concentration of water here now, so I'm not supposed to be underneath that, is it? Okay, so high water concentration here than it is here, so water will pass from this area here into there by diffusion. Larger molecules, e.g. the smaller proteins with molecular weights less than 69,000, are reabsorbed into the blood by endocytosis. Remember the process of endocytosis? Well, if you don't, look it up and just reinforce your memory, okay? Right, and there we have it. So, in this video we've described and explained production of urine with reference to processes of ultrafiltration and selective reabsorption. Now, in this video we're really focused on the what's happening in the glomerulus and Bowman's capsule, and also in the PCT, and in a subsequent video we'll look at what happens in the rest of the kidney. Okay, so thank you for watching, thank you for listening, and goodbye for now.